So good evening, everyone.
Good evening, sir.
Yeah, good evening. We are discussing about Mendel's laws. Yesterday we have discussed about the principles of inheritance put forward by Mendel. According to Mendel, there are four principles. There is principle of unit factors, which says that for every character there are a pair of factors which determine the contrasting traits of a single character. And that is what we call it as principle of unit factors. If the contrasting characters are involved, one of the two contrasting traits, when the pure line parents are said to be crossed, the character which appears in the F1 generation is called as dominant character, and the character which is suppressed is called as recessive character. And this we call it as principle of dominance. Okay, principle of dominance. And Mendel came to know when a tall plant is crossed with a dwarf plant in the parental generation, he could get all tall plants in the F1 generation. And in the F2 generation, he could get both tall as well as dwarf. That means the dwarf from the parent generation, the factor for dwarf character, passed on to the F1, which became hybrid because it carries capital T. That means one factor for tallness, the other factor for tallness. And during the formation of gametes, what Mendel says is that these two gametes are separated from each other. And this separation of factors responsible for a pair of contrasting characters in a F1 hybrid is called Mendel as segregation. So this is third principle of inheritance proposed by Mendel. And Mendel continued his experiments with a dihybrid cross. So in a dihybrid cross, he has taken two characters, seed shape, seed coat color. Seed shape is said to be round or wrinkled, and seed coat color is said to be yellow or green. So when an individual with the plant with yellow and rounded seeds is crossed with a plant with green and wrinkled seed, in the second generation, in addition to the parental combination of round and yellow and wrinkled and green, he got new combinations. What are they? Round and green and wrinkled and yellow. Why this happened? Because the genes for the two characters behaved independent of each other and separated or assorted freely so that new combinations in addition to the parental combinations are formed. And this is called as the principle of independent assortment. Okay, so thus Mendel proposed four principles or postulates, that is principle of unit factors, principle of dominance, principle of segregation, principle of independent assortment. And we, based on this, Correns and Shermark proposed two laws. And one is called as the law of segregation, and the other is called as the law of independent assortment. Okay. So now we are going to have a critical review of all the four principles proposed by Mendel. In addition, as these two are part of those principles, these are also included. Thus, we are having a critical review of Mendel's laws, in other words, Mendel's principles. Before going to that, let me explain you the reasons for Mendel's success first. Because there is one reason for Mendel's success, which is given, which is not by his research, but it is said to be, you can say, It's a godly gift. Okay. uh, for our success, we may attribute something to God even. So let's see what actually happened to that. So the first reason for Mendel's success is while all his predecessors considered many characters at a time, Mendel studied one character at a time. So this plant is having all the characters. It has seed shape character, seed coat color character, flower position, flower color, stem length, etc. and etc. But Mendel did not consider all other characters, but he has taken only one character for the study. And has seen how this character is inherited to the next generation. Had it been the fact that he included all the characters of the same plant and studied their inheritance, there will be no correct result because there will be the same plant will be having some dominant characters, some recessive characters. So he cannot conclude when all the characters are mixed together. So Mendel got succeeded only because of the fact that he studied only one character at a time. The second reason is because he is a mathematician as well as statistician, he has reviewed the entire process of inheritance with respect to the hybridization experiments and gave us a statistical analysis of the data. So, based on which we have seen in the previous slides in the last class, he has shown that there is 2.98 is to 1.01 like that. That means how scrupulously he has calculated the different kinds of uh, plants that are produced from the seeds. So because of this one only, he could come to certain conclusions. So this is the second reason for Mendel's success. And coming to the third reason is use of pure lines. So Pisum sativum, okay, he employed what we call self-pollination technique to get only pure line. So when we say this is pure tall or pure purple, what is meant by this? It has both the genes for tallness only. If it is for flower color, it has both the genes for purple color only. So when you study the inheritance of tallness, you have to ensure that both the genes are for tallness only. So Mendel, by self-pollination technique, he used only pure lines in the parental generation for his experiments. And while all other his predecessor continued their experiments only F2 generation. Someone stopped with F1 generation. Mendel continued his experiments till F3 generation. So, what is the result of this? Because he continued his experiments till F3 generation, he could know the genotype of the individuals of the F2 generation. In F2 generation, there are three tall and one dwarf. Everyone knows. But Mendel continued his experiments till third generation so that he could conclude that out of these three, one is pure tall, that is homozygous tall, and the other is hybrid tall, that is heterozygous tall. So based on this, he could arrive at the 1 is to 2 is to 1, which is called as the genotypic ratio. So the genotypic ratio of the F2 individuals is known only because he continued or carried his experiments till third generation. Okay. The next important reason for Mendel's success is complete dominance of selected traits. Correns, one of the three rediscoverers of Mendel's laws, he first time introduced the concept of incomplete dominance. That means out of the two contrasting traits, neither of the parental character appears in the next generation, but a mixing of these two characters appears. Thus, the tall character, for example, purple character and white character, let us take. So, when they are said to be crossed, According to Mendel, it gave rise only to purple character. But if incomplete dominance is involved, what happens? There will not be purple. Purple plus white, which may give rise to half half purple, that is intermediate purple or light purple color. Okay, but that did not happen with respect to any of the seven pairs of contrasting characters taken by Mendel for his experimentation, because all the seven pairs of contrasting characters exhibited complete dominance. Okay, so this is another God given gift for Mendel. And the next thing is testing of results with reciprocal crosses. So everyone before Mendel, they have crossed a male plant with a female plant. Okay, and they have seen how the characters were transmitted to the next generation. Nobody tested the influence of sex on the inheritance of characters. But what did Mendel do? He has taken the same tall and dwarf plants, but in the next cross, he reversed the sexes. In the first cross, the tall is said to be male and the dwarf is female. And in the next class, in the next cross, the tall is found to be female and the dwarf is found to be male. And in both the cases, the result is the same. And such crosses are called as reciprocal crosses. And by reciprocal crosses, he could conclude that there is no influence of sex on the Mendelian characters or characters that was studied by Mendel in Pisum sativum. Above all, the most important and the final reason for Mendel's success is the presence of factors for seven pairs of contrasting characters. So presence of factors for seven pairs of contrasting characters of P plant on different chromosomes. Mendel doesn't know anything about this. Later scientists have come to this conclusion. This is one of the reasons for Mendel's success. Why? According to them, if the factors or genes for the contrasting characters are present on same chromosome, what happens? Independent assortment will not take place. So Mendel could propose his fourth principle of inheritance, that is independent assortment, only because of the fact that the characters of the factors responsible for the seven pairs of contrasting characters are not present on the same chromosome, but rather they're present on different chromosomes. And let us say there are seven pairs of chromosomes in Pisum sativum. And we may think that if first chromosome, second, third, fourth, five, six, seven. So stem length character is present on one chromosome, purple flower on a flower color on one chromosome, flower position on one chromosome. Likewise, all the seven characters are distributed on the seven pairs of chromosomes. We may think so, but actually this is not the case. Later scientists, mainly a scientist called as Lamprecht, he observed that Lamprecht. Lamprecht observed that the genes for or the factors for seven pairs of Mendelian characters are distributed on only four chromosomes. They are first chromosome, fourth chromosome, fifth chromosome, and seventh chromosome. So factors for the seven pairs of contrasting characters selected by Mendel in Pisum sativum, according to Lamprecht, are present on only four chromosomes. That means one chromosome may contain one or two characters. The other chromosome may contain one or two characters. So here there are two, here there are two, four. And another should contain one, five, only one, six. That means another character is also present here. Thus, each chromosome contained more than one character. So each chromosome carries more than one character, factors for more than one character, which resulted in the proposal of independent assortment. But what the scientists also say that Mendel, for example, for seed shape and seed coat color, Mendel got independent assortment, but not for certain characters which we are going to see later. He could not get independent assortment, and he did not publish the results of those. Why? Because there is no independent assortment at all. Why that is so? Because they are present on the same chromosome. So, what is the mechanism behind this? We are going to discuss now. So, let us critically review the four Mendelian principles or the two Mendelian laws, Mendel's laws. Let us first discuss about the principle of unit factors. So, whenever someone says a plus b whole square is equal to a square plus 2ab plus b square. Okay, so this is the formula and this is the theory. And what it should apply? It should apply to everything. Then only it becomes universal. So whether it is 2 plus 2 whole square, minus 2 plus minus 2 whole square, or 100 plus 100 whole square, the same thing applies. That means that particular formula is universal. Likewise, Mendel says that for every character, there are a pair of unit factors. Okay? And we have to see whether it is universal phenomena or not. We can say, yes, it is universal. We can say, no, it is not universal. Why? Because unit factors which are in pairs, according to Mendel, are inherited from parents to offsprings in diploid individuals only. Pisum sativum, we know it is a diploid individual. Thus, there are a pair of chromosomes, and these pair of chromosomes carry two factors. Thus, one from the father, paternal, the other from the mother, maternal. So, for Mendel, this is universal. Why? Because it is universal to those individuals which are said to be diploid. So Mendel's statement is true because Pisum sativum is a diploid plant, but actually it is not universal. Okay, our answer is no. Why? Unit factors cannot be in pairs in plants or animals which are haploid. That means a haploid individual has only one set of chromosomes. A diploid individual has pairs of chromosomes, whereas a haploid individual has only one set of chromosomes. So in diploid individuals, there are two genes. That means paired. Whereas in haploid individuals, each chromosome carries only one gene. Either capital T it can carry or small t it can carry. Thus, where there is haploidy, there the principle of unit factors, which mentions that factors are in pairs, is said to be not universal. And at the same time, there is another characteristic feature that is polyploidy, another phenomenon called as polyploidy. So in haploid, there is only one set of chromosomes. In diploid, there are two sets of chromosomes. In polyploidy, there are more than two sets of chromosomes. Here we are representing three sets here. Okay. So in haploids, there is only one factor for a character. In diploids, there are two factors for a character as said by Mendel. Whereas for triploids, there are three factors present. For a tetraploid, if another chromosome is added, there are four factors present. So where there is haploidy and where there is polyploidy, principle of unit factors is not applicable. Thus, principle of unit factors as explained by Mendel is not a universal phenomenon. Okay, is it clear? Any doubts?
No, sir. No, sir.
Good. Second one, is principle of dominance a universal phenomenon? Okay. Before going to that, okay, we'll discuss about later. So, is principle of dominance a universal phenomenon? According to Mendel, out of a pair of contrasting characters, one suppresses the other completely. Then this is called as complete dominance, and one is dominant and the other is recessive. Mendel's second principle suggests complete dominance of one gene over other gene. We can refer it as factor also of an allelic pair. It holds good in majority of cases, including Pisum sativum, but it does not hold good where there are two phenomena. One is called as incomplete dominance, the other is called as codominance. Okay, so let us see what these are. Correns, for the first time, he has taken a plant called as 4 o'clock plant. This is the common name. Have to
Sir, your voice is breaking, sir.
Is it breaking for everyone?
Now it is
No.
okay.
Yes, sir.
Okay. So he has taken two varieties. One is said to be purple, the other is said to be white. So this is the parental generation. According to Mendel, what should happen? One of these two characters should appear. Either purple or white should appear. Okay. But here is one is red, the other is to be white in the parental generation. In the next generation, he got all flowers which are pink in color. And when the F1 individuals were intercrossed, what happened? The red, pink, and white characteristics in F2 generation appeared in 1 is to 2 is to 1. And this is the phenotypic ratio. Mendelian phenotypic ratio is 3 is to 1. Here, the phenotypic ratio is 1 is to 2 is to 1. Mendelian genotypic ratio is 1 is to 2 is to 1. Here also it is 1 is to 2 is to 1. So that means phenotypic ratio and genotypic ratios are same. So what happened here? Let us say this is capital R, this is small r, and this is capital R, small r. So what happened here? If this gene, capital R gene, is completely dominant over smaller gene. If the capital R gene is completely dominant over smaller gene, what happens? It should be red in color. But this capital R gene is having a reduced expression in the presence of smaller gene. So that the red color diminished to pink color. So this reduced expression says that capital R gene is not completely dominant over small r gene, rather it is incompletely dominant. And where there is incomplete dominance, there the principle of dominance as explained by Mendel, which is complete dominance, is not universally applicable. That is the first case. The second case is codominance. So co means together. Okay. So there is a mother or father with A blood group. There is a mother with B blood group. Okay. IB are the genes. When these got what we call married, they give rise to a child, okay, which is found to be having IA, IB genes. And here, what the scientists say is this one, the A group individual on the surface of RBC have all A antigens only. The B group individual on the surface of RBC have all B antigens only. whereas the child which is having IAIB genotype is having on the surface of RBC both A antigen as well as B antigen. Hence, her or his blood group is said to be AB blood group. So, father is said to be A group, mother is said to be B group, and both are homozygous. They gave rise to heterozygous child, which is AB group. And what happened here? Here, this is a heterozygote, IAIB. It is a heterozygous. On ninth chromosome, these genes are present. So there are a pair of factors in the F1 hybrid. This is the F1 hybrid. According to Mendel, one should suppress the other. But here, neither A suppressed B nor B suppressed A, but both are equally expressed. Okay? In IA, IB heterozygous condition, A expressed itself so that on the surface of the RBC, A antigen is present. B expressed itself so that on the surface of RBC, B antigen is also present. And such a dominance is called as codominance. So where there is incomplete dominance and codominance, Mendelian complete dominance is not universally applicable. Okay. In addition to these two dominances, dominance types, there are another two dominance types. One is called as pseudo-dominance and the other is called as epistatic dominance or epistasis, which we'll deal with in the coming classes. And the third one is said to be principle of segregation, which is called as the first law of Mendel or the law of segregation. According to law of segregation, what happens? Segregation of factors of an allelic pair is universally acceptable, which is the essence of genetics. So, what all the scientists say is in diploid individuals, segregation is a universal phenomenon because wherever there is meiosis involved, if there are a pair of chromosomes in me, when I produce gametes or when a plant produces gametes, one goes to this sperm, the other goes to this sperm or pollen grain. Likewise in female also. When the chromosome goes into the gamete, the chromosome which carrying the gene which is carried by the chromosome also goes into the same gamete. Thus, because there is meiosis involved, as chromosomes are separated, the genes carried by the chromosomes are also separated. So, thus, wherever there is sexual reproduction involving meiosis, segregation is a universal phenomenon. But it is not applicable to cases where there is non-disjunction involved. Non-disjunction involved. But non-disjunction is found to be a rare phenomenon. Non-disjunction is found to be a rare phenomenon. Hence, the scientists say that segregation is a universal phenomenon with respect to diploid individuals. We cannot consider haploid individuals for segregation. But for even polyploid individuals, there is segregation possible if they are even numbered for tetraploid, hexaploid, etc. For triploid and pentaploid, etc., there is another phenomenon that is involved there. Okay, let us see what actually non-disjunction, by which we are saying that law of segregation is not applicable and segregation is not possible. So, what happens actually? There are two phenomena: disjunction and non-disjunction. Dis means separation. Junction means where the chromatids are joined together, they are said to be separated. Then what is non-disjunction? Failure of separation. Failure of separation is called as non-disjunction. Separation is called as disjunction. So let me explain you this way. So we know we have chromosomes. I have a pair of chromosomes. This chromosome enters the S phase of cell cycle. G1, S, G2 we have now. So it enters the S phase. In S phase, what happens? DNA replication takes place. So this chromosome is having one DNA molecule. This chromosome is having another DNA molecule with each of which having its own centromere. So now what happens after the S phase? Each one undergoes duplication so that this one forms two DNA molecules and this one also forms two DNA molecules. Thus, I have now first chromosome represented by two DNA molecules along with histones. I call them as chromatids. And the second one is also represented by two DNA molecules, and that is called as again chromatids. So this is paternal chromosome, this is maternal chromosome. Okay, so the first pair of chromosomes are F after S phase are represented each by two chromatids. Previously, there is only one chromosome, one DNA molecule. Now I have two DNA molecules. That means two chromatids connected by a single centromere. Okay, so this is paternal chromosome with two chromatids and maternal chromosome with two chromatids. Now, after the G2 phase, it enters the M phase. In M phase, prophase, condensation of the chromosomes takes place. After that, it enters the metaphase. In metaphase, what happens? Spindle fibers are formed. Okay, with the centriole on either side. And now what happens? Each paternal and maternal chromosome are aligned side by side. So here paternal is there, a first chromosome, and here maternal first chromosome is there. Okay. And now what happens? Both are connected by the same centromere. Now this enters the anaphase. In anaphase, what happens? Both the centromere of these two chromatids separates. The centromere separates. Thus, there are two centromeres now. That means two chromatids are separated. So now what happens here is both the chromatids now separate from each other. Both the chromatids are separated and pulled towards the opposite poles. Thus, there is equal distribution of chromosomes in the cells that are produced from the parent cells. Thus, the daughter cells are having the same number or equal number of chromosomes. This separation of chromatids during anaphase 1 of meiosis 1 is called as disjunction. Okay, the separation of chromatids is called as disjunction. If what happens if there is no separation? Let's see. So this is how they are said to be. I'll remove everything. So this is how they are connected to the same centromere. Now, if separation takes place, there will be three chromatids becoming chromosomes coming onto one pole and the other to the other pole. That is disjunction. We call this as disjunction. If this does not undergo separation and these two have undergone separation, let us see what happens. What happens? Here, these two got separated, but there is no separation. Both the chromatids reach the same pole. The other cell, this cell will have only two chromosomes. This cell will have four chromosomes. Why? Because the chromatids fail to undergo disjunction. That means they fail to get separated. And this phenomenon is called as non-disjunction. So, in what way this is related to? In what way this is related to segregation? Segregation, we are saying that there are a pair of chromosomes, capital T, small t. What happens? One goes to the one pole, capital T is received. The other goes to the other pole, small t is received. Thus, we are saying that factors responsible for a pair of contrasting traits are separated. That means disjunction has taken place. Now, if these two don't get separated, if they come to the same chromosome, then what we call this as? This we call it as non-disjunction. When there is non-disjunction, there is no segregation at all. Both the factors may reach the same daughter cell, and thus where there is non-disjunction, segregation is not possible. Okay, and you have studied during your studies certain diseases, Down syndrome, okay, Edwards syndrome, okay, Klinefelter syndrome, Turner syndrome, Patau syndrome, and all these result due to non-disjunction. So when we discuss about the chromosomal mutations in the coming classes, then I'll be explaining you the importance of non-disjunction, sorry, the significance of non-disjunction, how it gave rise to the different diseases. If our ancestors say to us, every girl child every girl should be married at an early age. Okay, so when a female gets 18, the parents are said to be not letting her to drag the marriageable age. The maximum by 21, they let her get married. So it's some type of inconvenience for the girls to proceed in for further studies, isn't it? But actually, there is a reason behind this. There is a reason behind. There is a scientific reason for the early marriage of girl child. I'm not promoting this, but actually there is a scientific reason. When I explain you this, you will also agree to my terms. Not more than 30 or not more than 25. A girl should not wait for marriage. Why is that so? When we deal with developmental biology, I'll explain you that, or even during chromosomal aberrations, I'll explain you that. So thus, law of segregation, we can say it is a universal phenomenon, except for non-distinction, because it is a rare phenomenon, it is universally applicable. And coming to the last one, is independent assortment universally applicable or not? Okay, let us see. It is applicable to cases where the genes responsible for two contrasting characters. Okay, where the genes responsible for two contrasting characters, capital R, small r, capital Y, small y. That is the F1 hybrid, are present on different chromosomes. They are present on, first thing is, different chromosomes. That means, for example, this is the first pair where capital R, small r present. This is the second pair where capital Y, small y is present. So in this case, independent assortment will take place. Okay? It will not take place if they are present on the same chromosome. Independent assortment is not possible if they are present on the same chromosome. But there is another what we call exception. Even if they are present on the same chromosome, if they are present on different arms, this is the centromere, this is the short arm, and this is the long arm, small p and small q. If capital R and small r present on one arm, and capital Y and small y, sorry, just a minute. Okay. If capital R and small r are present on one arm, and capital Y and small y are present on the other arm, then independent assortment would take place. So, for independent to assortment to occur, there are two conditions. One is the genes responsible for the two pairs of contrasting characters should be present on the different chromosomes. Even if they are present on the same chromosome, they must be present on different chromosome. Okay, and on same chromosome and same arm, but are separated by longer distance. The third case is they are present on the same chromosome, capital R, small r, and capital Y and small y. But distance between these two should be longer enough. So that there are three conditions for independent assortment to occur. The first condition is both these pairs for two different characters should be present on different chromosomes. They should not be present on same chromosome. That is the first condition. For independent assortment to take place, even if they are present on the same chromosome, they must be present on different arms of the chromosome. Then independent assortment will take place. Even if they are present on not on different arms, but they are present on the same arm. Even if they are present on the same arm, they must be present at a long distance. Okay, they must be distantly located. So when these three conditions are present, we can say that independent assortment is possible. In other words, let us say in opposite. Independent assortment is not possible when the genes responsible for the two pairs of contrasting characters are present on the same chromosome. Independent assortment is not possible when they are present on the different chromosomes, different arms of the same chromosome, but crossing over did not take place. Okay, and if they are present on the same chromosome on the same side, that means same arm, if they are closely present, then independent assortment will not take place. So opposite to these three conditions, we can say that independent assortment is not possible at all. Okay, so let's see what this phenomenon is called as. So let us take one pair of chromosomes. I have capital R, small r, capital Y, small y. What happens during the formation of gametes? This one goes to one gamete, and this one goes to the other gamete. So now I have received capital R, capital Y, one gamete, and another gamete, small r, small y, another gamete. So what actually is here? Capital R, that is gene for round shape, is inherited together with the gene for yellow color. They are together because they are present on the same chromosome. So that what happens when these two get fertilized, there will be same capital R small r, capital Y small y. So the same parental combination is seen in the next generation even. There is no round and green combination being formed. Why? Because they are carried by the same chromosome. And this phenomenon is called as linkage. What is linkage? The genes present on the same chromosome are inherited together. The inheritance of genes present on the same chromosome together is called as linkage, and these genes are called as linked genes. If there is a phenomenon called as crossing over taking place, that means this chromosome is cut here, this chromosome is cut here, and both these segments are exchanged. Then what happens? Here comes small r, here comes capital R. Thus, I have a new combination, small r capital Y and capital R small y. That means wrinkled and yellow, round and green. That means even if they are present on the same chromosome, that means even if there is linkage, If crossing over takes place in same chromosome, if crossing, that means linkage is there, even if linkage is there, what should happen? If crossing over takes place, there will not be, there is every chance for the independent assortment to take place. But what the scientists say is, for crossing over to occur, the gene should never be present to side by side. If the gene for capital R and capital Y are present very nearer to each other, there is no endonuclease activity and the chromosome will not break in between these two because the distance is very less. So for the distance, if it is said to be increases, if the distance increases, crossing over may take place at this point. Breakage may take place and crossing over may occur. So crossing over to occur, the gene should be present on the same chromosome but at distant location. Then only independent assortment will occur. Okay, Mendel succeeded in postulating independent assortment because extensive linkage studies later conducted by a scientist, as I mentioned before, H. Lamprecht, demonstrated that 7 genes used by Mendel belong to 5 linkage groups. They are present on different chromosomes. What are they? They are present on first chromosome, fourth chromosome, fifth chromosome, and seventh chromosome. So it is obvious, therefore, that either Mendel did not conduct an experiment for independent assortment between certain combinations, as I've said before. Mendel excluded certain results because they are present on the same chromosome, they are closely linked. There is no crossing over occurring. Then what happened? There is no chance for independent assortment. Mendel deliberately might have set aside the results. So, for independent, Mendel did not conduct an experiment for independent assortment between certain combination, or else must have overlooked the lack of independent assortment between them, and he set aside the results, and he has shown us only the results where there is independent assortment. So, I'll show you. I will share you this particular what we call slide where which character is present on which chromosome, and that you have to be familiarized with. Seed color is said to be present on first chromosome. Seed coat and flower color is said to be present on first chromosome. And mature parts, smooth and wrinkled, that means inflated and constricted, they are present on fourth chromosome. Likewise, so on. You have to be familiar with all these characters. So what happens? Independent assortment is not possible when there is linkage because capital R, capital Y, and small y will be inherited together. Genes for seed shape and cotton color present on same chromosomes so that they are inherited together so that there is no recombinance, there is no independent assortment. That means when there is linkage, there is no independent assortment. When there is no linkage, that means they are present on different chromosomes. This is first chromosome, this is second chromosome. They are present on different chromosomes. Then what happens? There is every chance for the new combinations capital R, capital Y, small r, capital Y, capital R, small r, small y. Only this is possible only when they are present on different chromosomes. So Mendel's God-given gift is that the genes for responsible for certain characteristics in Pisum are present on different chromosomes. That is the reason why he could observe independent assortment. Okay, so that even if linkage is present, independent assortment is possible when there is crossing over. This is what you have to write down. So this is all with respect to the critical review of Mendel's laws. So any doubts, please ask me. Otherwise, we'll close here and we'll continue on the next topic, that is interaction of genes from tomorrow onwards. Thank you.
Thank you, sir.